Good evening, ma'am.
Good evening, sir.
Good evening, ma'am. Sir, you can start, sir.
Yes, ma'am. Thank you. Good evening, my dear students. You are all are once again welcome to the another biological session. So last class, what we have discussed already, you know very well what are the types of viruses also act as a vector. In this part, we were going to discuss other viruses used as a vector. What are the other types of viruses is there, already you know about cauliflower mosaic virus. Cauliflower mosaic virus. The disease name is called as mosaic. So what is the meaning of mosaic? A small white color board, taken examples, put more than one colors on it. What happened? It looked like a mosaic. It means different colors of patches present on the leaves. Particularly, a cauliflower mosaic virus means the cauliflower leaves also affected by this disease. The plants leaves or look like a mosaic nature. That's why they have given us a name is called as cauliflower mosaic virus. Okay. See, the simply called as CMV. It is one of the normally, about all the plant virus, it has a genetic nucleus, genetic material. Do you know which one? Dhanalakshmi. Good evening.
Good evening, sir.
Good evening. What about this cauliflower mosaic virus? It is a RNA virus or DNA virus, my dear?
Sir, not heard about it, sir.
You do not know. What about the TMV?
No, sir.
Tobacco mosaic virus?
No, sir.
Oh, anyhow. Okay, all the plant viruses, they have only the RNA as a genetic material, right? But in the cauliflower mosaic virus, it has a DNA as a genetic material. That is the difference. Just like a HIV, it is an exceptional case. For example, all the animal viruses, the genetic material is DNA. But taken examples of HIV, there is a RNA containing viruses, isn't it? Likewise, that all the plant viruses, it has RNA as a genetic virus. Particularly, the cauliflower mosaic virus is an exception. It has which one? A DNA as a genetic material. These are the differences when you are going to study the class or virus types of viruses or kinds of viruses. When we are going to study, that time we can identify. Okay, anyhow, cauliflower mosaic virus is a common virus used in plants because it affects the cauliflower. And herbaviruses and adenoviruses for human being as well as animals. Normally, but plant viruses also, it has a separate branches, and also animal viruses according to their disease. They may be divided into plant virus or animal virus. According to the nucleic acid, DNA virus or RNA virus. According to the structure, double standard or single standard or circular. Like there are three branches is there. One is the depends upon the plant or animal, which one affected. Okay. And then second one is depends upon the nucleic acid and then structure. Okay, anyhow. See, next one is the Gemini virus, paculovirus, retrovirus, TMB, otherwise called as tobacco mosaic virus. Here the cauliflower mosaic virus, here the tobacco mosaic virus. The tobacco mosaic virus is containing RNA as a genetic material, but in this category, cauliflower mosaic virus, it contains a DNA as a genetic material. Vaccinia M13, these are the other viruses used as a vector. It is also used for the genetic engineering and biotechnological process. Okay, see that one? See the next one. So this is considered as a cauliflower mosaic virus. The virus is nothing but the poison. Already you know who stated the statement. Virus is a poison was stated by the scientist Louis Pasteur. Okay. Any one of the virus, whatever, two components is very, very important. One is nucleic acid, another one is called as what is that? Protein molecule. The protein molecules may be capsid. It is outermost covering. Okay. Inside there is a nucleic acid is that whether it may be double stranded or single stranded. Suppose a single stranded means double S, small s, they have given DNA. Here what they have given, double stranded DNA molecule. This is the structure of double stranded DNA molecule. This double stranded DNA molecule surrounded by a covering layer is called as what is it, a capsid protein or virus associated protein molecules also there. It is sunken in nature. Sunken means it's also buried. Okay. It's also buried, partially buried like that. Okay. It's a sunken in nature, just like a stomata on leaves. Okay. So this is the structure of the cauliflower mosaic virus. Sometimes they may ask the questions in this part. Double stranded DNA. Cauliflower is a plant. The plant virus mostly found in which one, RNA virus. But in this category, it's a double stranded DNA molecule. Okay, see that one? The colmiviridae, virion, morphology. These are the two things that have been reported. One is called as isometric form, bacilloform, like that they have given. Isometric form means it's a root like a round. This one is bacillus means the structure look like a bacillus virus. Bacteria. Okay, these are the two types. This is also used for the, what is that? Vector. Used as a vector. See that this is considered a tobacco mosaic virus. The tobacco mosaic virus is nothing but look like a bunch of banana. Okay. The center there is a single stranded RNA molecule. It is very, very important, my dear. Single stranded RNA molecule present in it. It's look like a helical structure. Okay. At the same time, it is also covered by which one, the one of the layers called as capsid. Each and every capsid is also, each one is considered as a capsomere. More than one capsomere united together form as a capsid. So the capsomere is nothing but the unit of the TMV, tobacco mosaic virus. Okay. More than one capsomere united together form as a capsid. It's just like a barrel-like structure. See that one? A bunch of banana. See the next slide, you can easily understand. So this is the structure of the tobacco mosaic virus. The center there is a RNA molecule is there. It is also coiled in nature. Okay. The RNA molecule covered by the protein molecules. That protein molecule is called as what is that? Capsid. Suppose take an example of one capsid is one unit is called as capsomere. It is nothing but the protein molecule. Okay. Don't forget, my dear. This is very, very important. This was introduced by tobacco mosaic virus was discovered by the name of the scientist. Anybody else? Dhanalakshmi, do you know who discovered tobacco mosaic virus?
No, sir.
You don't know? Okay, anyhow, please understand. Okay. There are three scientists who have been reported. First one, and second one, third one, who discovered it? What about the V.M. Stanley? V.M. Stanley. Who said the contagion VVM flu? Who discovered the tobacco mosaic virus? These three part is very, very important, my dear students. Okay. So I will tell you next. Okay, see that one. Next, the bacteriophage vectors. Already we discussed bacteriophages. Phages is nothing but the virus. It's act or infect the bacteria. Is such type of viruses are called as bacteriophages. Or viruses that infect bacteria. These are usually called as phages. Phages can be double standard, which one or single standard. Suppose if it is double standard, it is also denoted T phages. T phages means it's nothing but the numbers. T2, T4, T6. These are the numbers. Okay. So double standard molecules. Single strand. There are both DNA and RNA. See, T2, T4, T6 or DNA, and RNA is MS2. Okay, phages. These are the phages. Different types of phages are also reported. It's also act as a vector. Okay, next one is the major advantage of the phages vector is that while plasmid accept DNA pieces about 6 to 10 kilobase long, phages can accept a DNA fragment 10 to 20 kb long also. This is the maximum size of the acceptor. How many kilobase that can easily absorbed by the vector, that is very, very important, my dear. Okay, so this is very, very important. See, the next one is bacteriophage is a lambda phage. There are so many phages have been reported, particularly T series, T2, T4, and T6. Okay, and another type of the bacteriophage is also used where in the genetic engineering and biotechnology as a vector. So that one is called as what is known bacteriophage lambda phage. So lambda. It has a double stranded linear DNA genome of. It has a double stranded linear DNA model. Don't forget, my dear. Linear. Sometimes they may ask the question, which one of the following is a linear structure of the DNA molecule present in it, otherwise called as linear DNA genome present in it. Like that, they may ask the question. Okay. So, how many base pair present in it? See here, 48.514. That is denoted as a 48,514 base pair. Okay. So, in which the 12 bases at each end are unpaired but complementary. That is very, very important. This is also another important point. Okay. This is the another one. So, in which the 12 bases at the end or unpaired, it is not complementary with each other, but at the same time, it can be easily complementary with the opposite strand. Okay, this end are therefore sticky or cohesive and are referred to as the cos site, cohesive end site. This is very, very important, my dear students. Sometimes they were asked the question in bacteriophage, the lambda phage. Okay. The end, sticky or cohesive end. What is the meaning of sticky or cohesive end? Just imagine when the restriction enzymes cut the DNA molecule, that time what happened? Automatically, you will get the two end, isn't it? That is what is a polyandromic gene, where there is a polyandromic gene present in it. That area will be cut, make a cut by the molecular scissor. That is called as molecular scissor restriction enzyme. Okay, that site is which one? The cohesive. So, what is the meaning of cohesive? I will explain it later. Okay, after this, how it will be. These sites are important for packaging DNA into phage heads. This site is very, very important. When you are going to study the lambda phage genome structure, that time you can easily identify, understand cos side, which area is cos side, and that which side is that. There are two sides there. Bacteriophage lambda genome is also divided into two sides. One is called as cos side, otherwise called as head side. Another opposite side is also that is called as tail side. We'll see. Okay. So 1/3 of lambda gene is non-essential and could be replaced with foreign DNA. 1/3 of the DNA molecule is non-essential and could be replaced with the foreign DNA because this genes is not represent any replication process or protein molecules. So unwantedly present like that, non-essential. There is no need, whether present or absent, no need, that one. There is no any properties for the particular genome. That genome, that's why it is called as lambda genome. Okay, it can be easily replaced by the foreign DNA molecule. That is very, very important. Okay. See that one. You can see the structure, you can understand. So this is the structure of the which one, lambda phage. That base pair you must know 48,514 base pair. This is it. Okay. Center 851 you put and both the side 4 is adapted. Okay. 48, 51, 4. Whatever it may be. You can easily identify. Okay. So these are the age, these are the structures. Here there is divided into two types. One is head side and tail side. The tail side, there is a cos side is that COS. The head side, there is a ATT. Okay, this is the, and then see the tail sides. Okay, see that this is the portion of the lysis, this is the replication portion, and regulation portion is also the recombinant portion. They may be divided into three parts according to their structural aspects, and the functional aspect they may be divided recombinations, regulation, and replications. Finally, lysis, it occurs. Okay, this is the tail part, this is the head part. Okay, see the next one. So lambda genome. This is also a lambda genome. See that one? Lambda genome only. Genetic map. Genetic map look like a this. Already we studied that thing. So genetic map used for the crossing over. It is a graphical representative of the gene. Any one of the gene, whether plant or animals, we can use it a genetic map, which nucleotide present, which location we can easily identify. And also the crossing over was also very easy. Okay, Morgan, the centimorgan already studied. The gene map is also measured by the unit is called a centimorgan. Already you know about that things also, isn't it? Okay, see the next one. So this is also another lambda gene, bacteriophage, lambda. Each one it has a different numbers also. See that lambda page number has given here 48514, isn't it? Sometimes 500, 512. Like the different base pairs also depends upon their nitrogen base pairing. Okay. Now we are going to see that what is by sticky end, what is by blend end. Say this is the DNA structure. Okay. After the DNA structures undergoes the what is the digest? What is the meaning of digest? When you are adding the enzyme restriction enzymes, the cut made like this. Cut made like this. Here there is a one base pair is what is a head left here. Here also one. So these type of what is that? Divisions. This is considered as a sticky end. It can be easily bind with this one. This is also sticky end. This can be easily bind with this one. Here, this type of division is called a sticky end. It's present mostly in the E. coli. E. coli, Escherichia coli restriction 1. But here, blend end E. core 5. See that one? So same is also present in it. Digest, you will get which one you know. There is no any free nitrogen base. So see here. Both are having a blend end. This is considered as this type of separation of the DNA molecule is considered as a blend end. This is considered as a sticky end. Okay, both are same genomes present in it. Okay, but this the structure is different. See the E. coli 5, so E. coli 1. Okay, when you're adding the Escherichia coli restriction enzyme 1, when we are adding, we will get the sticky end. When you are adding the 5, equal 5, Escherichia coli restriction enzyme 5, what happened? We will not get the sticky end, we will get the blend end. There is no any, what is that? This type of single base pair, you will not get it. This structure is called as polyandrome. Already you know about it. Both the side you will read. So when you are reading this 5-N to 3-N, 3-N to 5-N, what happened? You know, the same nitrogen bases present. You know about Malayalam. When you are reading the opposite manner is also, we can read Malayalam also. Okay, that is very, very important. See that one? I will show you. So then only it will be better for your understanding. Okay. See, so this is a structure. This is the 5' and 3'. This is considered a 5'. So this is the 3'. Here opposite already in our elbow. This one is 5'. This one is 3'. Okay. Here the nitrogen base is there. Already you know very well. Okay. How many numbers we cannot able to. Any fragments it has a complementary genes according to their nitrogen bases. Suppose if it is adenine here, so there is a thymine here, isn't it? So thymine, guanine, cytosine, like that, isn't it? But the polyandromic already I told you. So it made a cut in this position. In this position, you will get this structure. There is a sticky end like this. Okay, this is a sticky end. So this portion is called as what is a sticky end. Sticky end. Okay, this is a sticky end. Okay, this is what happened now when you are adding the which enzymes. Ecorib 1, eco Escherichia coli. Which one, restriction enzyme 1. Don't forget, my dear, this is a very, very important 1. So now that time what happened, you will get the sticky and like stick. Suppose if it is the same process, when you're adding the E. coli 5, what happened now? You will not get the sticky end, you will get the structure look like this one. The both are same like structure. You know the difference between these two? This is the sticky end, isn't it? This portion is called as what is it? Sticky end. Okay. So you will not get the sticky end. What is the meaning of sticky end? It's equally divided. Okay. There is no any free nitrogen bases present in it. That is very, very important. Okay. Here, blend end. This is considered as a blend. Okay. Blend end. Okay. Don't forget, my dear. This is very, very important. See, go to the next one. See, the same structure has given here. See, this one is a sticky end, this one is a blendy end. Okay. See here. So, which enzyme is responsible for that? E. coli R1, eco R1, E. coli 5. Okay, that is very important. See, the next one is also similar. See that blendy end. When you are make a cut with the help of the molecular scissor restriction enzyme, E. coli 1, what happened? This one we will get 5. You will get like this, isn't it? There is no any free nitrogen bases. Here also the restriction enzyme, it can cut any position, the polyandromic gene. Okay, we will get the like this. This is called a sticky end. This one is called a sticky end. This is the sticky end. Okay, so go to the next one. See that lambda phage structure is also. The lambda phage structure, see that hexagonal. No, it is not a hexagonal. It's a different types is there. Okay, head containing DNA molecule adeno, but covered with the protein molecule. There is a collar is there, and also the body part, and the tail is also there. Okay, tail part is also having a binding protein molecules. It can easily adhere on the surface of the any bacteria. It can easily fix it. That is the tail plate. Base plate is also there. It can easily bind with the bacteria. That's why it is called as a what is that? Lambda phage. But at the same time, what happened? No, the DNA molecule. What happened? Some of the genes it is not responsible for the which one non-essential one. Okay, some of the gene is not essential functions. Okay, so it can be easily replaced by the which one foreign DNA molecules. Okay, I think this part is completely finished, my dear. The question part. See the first question. Okay, see in gel electrophoresis, separated DNA fragment can be visualized with the help of which one is correct, my dear? Dhanalakshmi.
Sir, ethidium bromide.
Ultraviolet radiation.
UV radiation.
Right, very good. Exactly correct, my dear. Okay. So go to the next one. The DNA fragments are which type? Neutral, negatively charged, positively charged, and either positively or negatively charged, depend on their size.
Sir, neutral.
No. Here you just imagine, my dear, DNA fragments. It is not DNA. DNA fragments. When you will get it this one. After the electrophoresis, gel electrophoresis, you will get the small fragment like structure. The fragment containing which charge? Maximum?
Negatively charged.
Yeah, negatively charged. Don't forget, my dear. So this is very, very important. And then third one is the process of separation and purification of the expressed protein before marketing is called as which stream? Already we studied these things also. Upstream processing.
The downstream processing.
Very good. Downstream processing. Okay. And then the electroporation procedure involves the fast passages of food through C-pores in phloem elements with the help of the electric stimulation, making transition pores in the cell membrane to introduce the gene construct, opening up stomatal pores during night by artificial light, electroporation, don't forget. Purification of saline water with the help of the membrane system. Which one is correct here, my dear? Which one?
Sir, option 4.
For purification of saline water with the help of the membrane system, electroporation. Is it correct, my dear? Is it correct?
Sir, doubt, sir.
Okay, anyhow. So the making transition pore in the cell, see that poration, isn't it? Pores in the cell membrane to introduce gene construct. So this is electroporation. Now only we studied, isn't it? Before 1 or 2 classes before we studied electroporations. Gene constructed methodology only the electroporation mostly used. Okay, other things are not coming. See that plant stomatal is not possible. Purification saline water with the help of the membrane system. No. So this is the correct one. So go to the next one. The agarose extract from the seaweeds finds use in tissue culture, PCR, gel electrophoresis, and also spectrophotometry. Agarose extract from the seaweeds finds use in
Gel electrophoresis, sir.
yes, given the answer, isn't it? Gel electrophoresis. Very good, because it is related to our topic. Gel electrophoresis. And the last one, which of the following is the correct sequence of step in the PCR polymerase chain reactions? Denaturation, annealing, and extension. Denaturation, extension, and annealing. Extension, denaturation, annealing. Annealing, denaturation, and extension. First of all, you should know which one is happened. Which one happened during PCR? Which step is involved? There are 3 steps is there. Same process, annealing, denaturation, extension. Isn't it? Which one taking first place? First of all, there is a double-stranded DNA molecule. You have to split the molecule. So splitting means which one is come first? Denaturation. Then cooling. After that, extension. So the first one is the correct one. Okay. Which one is the correct one, my dear?
The denaturation, annealing, extension.
Yes, this is the first. This is the correct step. Steps involved in the PCR. Okay. Don't forget, my dears. So today what we discussed, this is the things. Okay. Exactly, we made a cut after the molecular scissors. We have to select the foreign particles of the other organism and replaced it. Here there is a 4 step they were used just for the image only, but we cannot able to like this. Okay, they were used the molecular scissor, the ligase enzyme used for this purpose, linking process. Cutting process, what they were used? No restriction enzymes. Which area? That is origin area, the polyandronic genes. Okay, they made a cut sticky end. Okay, all those things happen inside. Okay. Okay. So just we saw it. What are the things? Just revise it, and then we'll pass on to the next lesson. Today, what we have discussed, the cauliflower mosaics, other viruses also used for the vector process in genetic engineering, particularly cauliflower mosaic virus is there. Okay. What is the structure related? It is nothing but the double-stranded DNA molecule surrounded by the capsid protein molecule is also there. And also tobacco mosaic virus, it is one of the RNA containing plant viruses. A single stranded RNA molecule center, it's occupied, surrounded by the capsid is there. That is nothing but the protein layer is called as capsid, and that each one is considered a capsomere. It's nothing but the unit of protein molecule is called as capsomere. Okay. So, and then next one is the bacteriophage vector. Bacteriophage vector is many things have been reported. They are all considered getting known as double standard T series bacteriophages, T2, T4, T5, T6, up to so many things is there. Depends upon their molecular size and structures. Okay. And also one more is that single standard. Okay, bacteriophage is also present in it. Both the DNA and RNA bacteriophage is also present, and particularly MS2 is the RNA phages. maximum amount of the plasmid accepted by the DNA molecule, how much, the 6 to 10 kilobase long, and also the fragments 10 to 20 kilobase long. This is very, very important, my dear. Next one is lambda phages. Lambda phages is nothing but the linear DNA genome present in it. It is also one of the bacteriophage. It's also a lambda phage. They have given as a lambda phage. Okay, it contains the linear DNA genome and the molecular structure. How much, 48,514 base pairs. From this one, each in which the 12 base pair at each end, the unpaired but complementary in nature. These ends are therefore sticky or cohesive and also referred to as the cos site. Which site? This is very, very important. Sine, cos, like that. Cos site. Simply cos means why they are called as cos site. Cohesive end sites. That is called as cos sites. These sites are important for the packaging of DNA into the page head. 1/3 of lambda genome is non-essential. 1/3, 1/3. Okay, and could be replaced with the foreign DNA molecules. Okay, this is the structures. See that one? So this is the structure of the bacteriophage, 48,514 base pairs. Okay, here it may be divided into head and tail, and also what happened, the recombination gene portion, regulation portion, and replication portion is also lysis. These are the entire structures, okay, of the lambda phage. Next one is also same, lambda genome. Okay, so same things. It has a specific structures. Okay, and then there are what is the difference between sticky end and the blend end? Sticky end is also done by the E. coli or 1 restriction enzyme 1. Okay, at that time what happened? You will get the a structure look like this one. Okay, there is a polyandromic. Already we discussed so many things is there. And then at same time blend end, how it will be obtained? E. coli 5. So you will get the blend end look like this one. So this is the sticky end. This is the, because here the free nitrogen base is there. Here there is no nitrogen base freely obtained. So such type of separation is called as what is it? Sticky end, blended end. Okay. So the same things is also repeated here also. Okay. This is the lambda phage. And what are the questions already we discussed that thing? Already we know all about it. Okay. We will pass on to the next one is part 3. Okay. See. Next important, my dear students. We'll pass on the next one, the part 3, biotechnology. So this is also very, very important. What are the things he has given here, in this part, transposons. Okay. So many times they were asked the question, what is meant by transposons? Sometimes they may ask the questions, jumping genes, transport from one place to another place. Jumping means one place to another place, whatever it may be. One location to another location, like that. Okay, see that one. The transposons class of genetic elements that can jump to different locations within the genome. That is very, very important. It is not present anywhere. It will not present in a particular point. It can jump any locations. Such type of gene is called as jumping gene, otherwise called as transposon. In other word, we can call which one, transposons also called as jumping genes. Okay. Although these elements are frequently called jumping genes, they are always maintained in an integrated site in the genome. So in jumping gene is also present, but it can move from one place to another place, the 5th place to 10th place, 10th place to 100th place, whatever it may, but at the same time, it maintains an integrated site. There is no any differences. The genome aspect, genome level. In addition, most transposon eventually become inactive and no longer move. This is very, very important. Okay, see that's one. There are jumping genes that often change their position in the genome. Transpose can be converted into vectors. Okay, the transport gene is also used as a vectors. Okay, so what are the transport genes are present? One is called as cosmids. What about the plasmids? But here, cosmids, they give a name is called as cosmids. What is by cosmid? Who discovered cosmid? Where it will be act? See that cosmid is a first described by Collins and Horn in 1978. It's a type of excuse me, hybrid plasmids with the bacterial ori sequence, a cos sequence derived from the lambda phage. Already we discussed. Which one is considered as a cos site? Ori means already in our board, where there is a replication process started. That place is called as what is that? Ori, isn't it? So here the cosmid is also called as what is a jumping genes. Cosmid also called as a jumping genes. This was first discovered by the name of the scientist Collins and Cosmid Collins. Don't forget, Collins and Horns in 1978. It's a type of hybrid plasmids. Hybrid means, but the combination of two. The bacterial ori sequences of cos sequences derived from the lambda phase. Here the bacteria is ori and the cos is the lambda. So these are the two combinations. That's why it is considered as a hybrid. Bacteria as well as the phage. These combinations. See the transposable elements. See, so this is the structures. Okay. So this is the transposable elements. It can be easily transferable. Okay. So this is the structures. Flanking direct repeats and the terminal inverted repeats, like that they told. They can jump any places. What happened in the gene sequence, there is no any variations occurs. Such type of genes are called as what is a transpons. But at the same time, it will not indicate any protein molecules. See that. Cosmid vectors are its types. So this is considered as a cosmid vector. Already we studied. Here there is a two site is there. One is called as ori site and cos site. The cos site is also obtained from where? The ori is also obtained from where? Ori is obtained from where? The bacteria, and cos site obtained from the lambda phage. Already know about. Amphicellular resistance. This is also already present in plasmid and cloning site. These are the things. These are the entire structure of cosmid vectors. We can replacing where? In this area, anything, whatever it may be. Which one, if you want to select, I think it can be easily replaced in this area, only cloning site. Okay, hybridization also takes place in this part. This is the structure of the cosmid vector. Okay, see that. The cos site is a sequence required by a DNA molecule in order to be recognized as a lambda genome. Already I told you, cos site, lambda genome. By the protein that package DNA into lambda phage particles. Okay, the cos sites, there is a lambda genome is there. The lambda genome protein and also that package DNA into lambda phage particle, collectively called as lambda phage particles. These vectors provide an efficient means of cloning large fragment of the foreign DNA, much more than a vector, and can accommodate, not in the single one. It can accommodate a large fragment of foreign DNA molecule. It has the ability to carry more than more number of what is that? Fragments, foreign DNA, much more than a vector can accommodate. It can be received. That ability it has, that is called as cos site. That position is called as cos site. Okay, don't forget. And also phage meet, another one of the phage meet cloning vector. Phage means already know about. It is nothing but the bacteria affecting virus is called as phage. Meet means the isolous plasmids. So they may add the both word, first letter and last letter. Plasmid, the last letter. Bacteriophage, phage meet. Okay. Our cloning vectors developed as a hybrid of the filamentous phage M13. Don't forget the phage meet is also the one of the vector developed as a hybrid of the filamentous phage M13. That is very, very important. M13. And plasmid to produce a vector that can not only get back as a phage particle, but also can propagate as a plasmid. Example, B blue script 2Ks. This is the another one. How it will be? The M13, right? Okay. It is one of the hybrid vector, bacteria as well as the phage. Okay. The plasmid to produce a vector that cannot only get packed as a phage particle, but also can propagate as a plasmid. It is not a phage particles. Phage particles mean, you know, but it's nothing but the nucleic acid. But at the same time, it also acts as a plasmid, propagated as a plasmids only. So, say example has given is a plasmid blue script 2Ks. The carrying capacity of the phage meat is higher than phage vector. This is the difference between phage meat as well as a phage vector. Okay, do you understand here? Here, plasmid. Okay, lambda particle, phage particle. Here, the same things is there, but at the same time, hybrid, M13 hybrids of the plasmids. Okay, how it will be act as, you know, it act as a, what is that? Plasmid, propagate as a plasmid. It is not a phage particle. Example, B plus script and 2Ks. That is very, very important. The phage meats are genetically more stable than recombinant phage vectors. One of the first hybrid phage meat vector was PEMBI, constructed in the year 1983. EMBI. Okay, the plasmid EMBI is the first hybrid phage meat. Don't forget, this is also very, very important. Sometimes they were asked the question in this part. Which hybrid phage meat is vector was first discovered, first constructed, first discovered, like that they may ask the question. Okay, see that one? EMPI. Okay, don't forget, mate. Go to the next one. See that EMPI, the blue script 2SK structure. The lag jet is also there. MCS, AMP are resistant, and ORI. ORI already know about. ORI comes from where, you know? Bacteria. Ampicillary resistant already. Lag jet. It is also one of the gene already present. MCS. This is, see the base pair, the blue script 2SK base pair is 2961 base pair. Sometimes they may ask the question in the blue script, always called as phage meats. Okay, given examples. Which one of the following is the examples of phage meat, like that they may ask the question. Sometimes the phage meat containing, particularly the blue script 2SK containing which, how many number of base pair? Okay, like that they may ask the questions. Okay, so 2961. 2961. Okay, so this is the artificial chromosomes. The last one is called as what is an artificial chromosome. Artificial means man-made chromosome. We can construct our own gene, like as a chromosomes. The artificial chromosome are DNA molecules assembled in vitro. This is very well. Form defined constitution that can function like a natural chromosomes. The artificial chromosome also, what is that? Can function like a natural chromosomes because the structure is constructed by the lab. Man-made chromosome is called as artificial chromosomes, but it behave like as a natural chromosomes, mostly done in the within the lab, in vitro condition only. Get it? Okay. See, the concept booster is just noted on other the types of artificial chromosome given in your book. This is very, very important. So many times they were asked the question, what is my BAC? What is my YAC? What is my MAC? BAC means bacterial artificial chromosome. BAC. ABCA may be written as a BAC. OAC, MAC. Y means ESD. M means mammalian artificial chromosomes. These are the three things have been given. Okay. The bacterial chromosome means what is that? It's isolated from bacteria. Which bacteria? See that one. The BAC is a cloning vector system in Escherichia coli developed by the Mel Simon. Sometimes we ask the question, the scientist name. The bacterial artificial chromosome BACs are designed for the cloning of large DNA insert in E. coli host. Already you know about all the cloning process which occurs within the E. coli only. Anywhere, any places. E. coli is act as a very good and super and coast. Okay, selected by the scientist. So the first one is called the bacterial artificial chromosome. Which one? It's a vector system in E. coli. Who developed? Melson and Simon. Okay, that is the reactions. And second one is the yeast artificial chromosomes. Yeast artificial chromosome is also called as YAC, Y-A-C. It was first described in 1983 by Murray and Sostak. YAC vector can accommodate 100 to 500 kilobase of insert DNA. Normally, about 10 to 20 kilobase per only. Okay, but in this category, the yeast artificial chromosome, they can accept or accommodate 100 to 500 kilobase per of the inserted DNA molecule. This is a important one compared to the other two. Okay, and the last one is called as a mammalian artificial chromosome, simply called as MAC. The MACs or mammalian artificial chromosome, like a YOX, rely on the presence of centromeric and telomeric sequence and origin of DNA replication. So these are the types of artificial chromosome. Artificial means man-made. They have chosen three things. One is called as bacteria, is animal. Yeast, so fungi. Mammalian, animal. Okay, likewise, they were selected. Here you must note it down. Bacteria means which bacterium? Yeast already know. Saccharomyces cerevisiae. Okay, so Mireya and Sostak, 1983. So already you know the 1983 also comes where see that one? 1983. See that? Phage meat is also discovered in the year, which one 1983. Don't forget, my dear. So comparison between the things. Mammalians means human beings. Artificial chromosome like YOC. So this is all the same. Okay. But in which stage present in the centromeric and telomeric sequence and the origin of the DNA replications also takes place. These are all considered that is known as types of artificial chromosomes. FOC, YOC, MAC, already studied. And this HACs also there, human artificial chromosome. PACs also there. P1 derived artificial chromosome. These are the other two things is there. These three only given in a book, PAC, YOC, and MAC. Bacterial artificial chromosome, yeast artificial chromosome, mammalian artificial chromosomes. Okay. HACs, human artificial chromosome. P1 derived artificial chromosome also given. See that vector insert size. This is also very, very important, my dear. Please note it down, my dear. Plasmid, more than 10 kb size, we can insert plasmid. Okay. Suppose if it is bacteriophage, 9 to 15 kb. Already we discussed 10 to 20, isn't it? In between. Okay. Casmids, use note it down, my dear. 23 to 46. Just double it. 23 double it to 46. Isn't it? In between that range. Don't forget casmid. So the casmid is nothing but the 23 to 45 kb. PAC, bacterial artificial chromosomes. See that? Equal to 300 kb or more than 300 kb kilobase per. PAC, 100 to 300 kilobase. And YOX, 100 to 3000 kb. So this is very, very important. Which one is the highest here? YOC is the highest here, isn't it? Don't forget, my dear. This is very, very important. So which one of the following it has a more genetic material or gene inserted size? Like that, they may ask the question. YOC is the highest value of the inserted sites. This is also very, very important, my dear. Okay. And the next one is marker genes. Okay. So we will see the marker genes on the next classes. Today, we will summarize all those things. What are the things already we discussed? See that one? We will finish up to this level only. What is my transposons? Transposons is nothing but the one of the a genetic element that can jump to different locations within the genomes. Okay. Although these elements are frequently called a jumping gene, they are always maintained in the integrity site in the genomes. That is very important. In addition, most transposon events become inactive and no longer move. This is also very, very important. Sometimes they may ask the questions, what is a assessment reasoning? Okay, which one of the following genes it is inactive and no longer move? Okay, like that they may ask the questions. So just note it down: transport genes, plasmids, and vector. Like that they may ask the questions. You have to exactly give the answer. Jumping genes, otherwise called as transposons. Okay, that is very, very important. Their jumping genes are that often their position in their genome, the transposon can be converted into vectors. What are the examples? Cosmid is also one of the examples. This was first discovered by Collins and Horn in the year 1978. It has a two-part. One is hybrid because it's hybrid varieties. Okay, the bacterial ori, sequence of cos sequence derived from the lambda phage. One is a virus, another one is a, what is that? Bacterial cosmids. So which area? Ori is a gene. Cos is also one of the site, otherwise called is also one of the part only sequences derived from the two different microorganisms. So one is bacteriophage, another one is called as a bacteria. Which one? Ori is bacteria, cos is a phage. Combined together, you will get the cosmids. Okay, that is very, very important. The transport already given in there. This is the structure of the transport segments. The cosmid vectors, already I told you, this is the cos site and ori site. Ori site bacteria, cosmid site, which one you know? The lambda phage. This is the cloning site. Cloning site, we can add a number of, what is that? Foreign genes. It easily accept. Ampicillin resistant site is also there. Plasmids already in our tetracycline as well as a ampicillin resistance already present in it. And cos site is the sequence recorded by the DNA molecule in order to be recognized as a lambda genome. DNA into lambda gene particle because it's nothing but the protein encoder. Okay, these vectors provide an efficient means of cloning large fragments of the foreign DNA. That is very, very large fragments means number of fragments 100, 200, 1000, whatever it may be. How much we can possible, which can be possible, right? That can be accepted by the DNA molecule. Such type easily accommodated, the vector also. Such site is called as cos site. That gene is called as lambda genome. Known for cosmids. And phage meet already in our lab, it's a combination of the two filamentous phage of the M13, filamentous phage. The name is called as, the example is called as a B blue scripts, a plasmid blue script at 2Ks. Okay, 2Ks. Already we saw it, that things also. Which one is the example? See that one? BEMP1. This is also another one. See that one? This one, blue script 2Ks. But here they have given another example. Okay. Because it is the first discovered or first identified, produced in the laboratory aspect, PEMB1, constructed in the year 1983. Okay. See, just EMP pathway already studied in glycolysis, isn't it? Embeden-Meyer-Parnas pathway. Just like that, you can imagine or remember plasmid, Embeden-Meyer-Parnas, like that, just okay, just for your examination point of view, identification only. Okay, but it has a specific name, plasmid EMPI, constructed in the year 1983, just like a pacemid. See the structure, the blue script. So 2961 base pair is the, okay, so already know our artificial chromosome. It is also similar to that of the natural chromosomes. Okay, it is also constructed within the lab in vitro, and also define constitution that can be function like the natural chromosomes. That is also very, very important. And the last one is called concept booster. Particularly, you must note that there are so many artificial chromosomes have been reported, particularly the bacterial artificial chromosome, simply called as PAC, the yeast artificial chromosome YAC, and the mammalian artificial chromosome is called as, what is that? MAC. Here the E. coli, here the yeast, and here the mammalians. The bacterial artificial chromosome is called as, where will you accept? Where did you get it? Escherichia coli. Okay. Mostly they prepare the bacteria. Which one you know? Escherichia coli is genetic engineering or whatever it may be. Yeast artificial chromosome. This was first described in the year 1983. Same one as it is a PageMat and also EMPU also. Murray and also Sostak. How many base pair it can be possible? Already we studied in the tabulation for 3000 kp, isn't it? But in this category, 100 to 500 kp have been given. Mammalian artificial chromosome is what is a MAC. Okay, it is similar to that of the yak, but here which position, presence of centromeric and telomeric sequence and the origins of the DNA replications takes place. Okay, see that one. Types of artificial chromosome: PAC, yak, MAC, HAC, PAC. BAC's bacterial artificial chromosome, Escherichia coli, yeast already know about artificial chromosome. MAC's mammalian artificial chromosome, HAC's human artificial chromosome, PAC's and P1 derived artificial chromosomes. How many size they can be inserted? Vector, these are the vectors: plasmid, bacteriophage, cosmid, PAC, and PAC and BAC. Okay, yak. These are the plasmids. How many size which can be inserted in the foreign particle? Like as a foreign particle, see that one. This is considered as more than 10 kp. This is maximum 9 to 15 kp, just like a 20 kp. Okay, 23, 46, 5 kp in cosmids. PAC 300. PACs also 300, up to 300. The yaks, just imagine 100 to 3000 kp. Which one is the highest? Which one is the lowest? These two is very, very important, my dear. Okay. So that's all about today class, my dear students. We will see the remaining things on the next classes. Thank you so much for the participation and take care. Bye. Good night. Thank you. Bye.
Thank you, sir.
Thank you. Bye. Take care. Thank you, ma'am. Thank you so much, ma'am.